All right, so chapter 23 is going to focus on the evolution of populations. Um, a common misconception regarding evolution is that organisms are able to evolve throughout their lifetime. That is not the case. Natural selection does act on individuals, but it is their populations that evolve, not the individuals. So here there was a population of finches on Daphne Major Island. There was a drought, and the larger beaked birds were more capable of survival because they could crack the larger seeds. Because they were able to survive and reproduce, their alleles for the large beaks were passed on, and the finch population did evolve as a result of that change to their environment. So we talked briefly in class. Neoevolution or microevolution is more specifically addressing the changes in allele frequencies that occur over generations within a population. Not all of the sources of these allele frequency changes are going to be a result of adapting to an environment. The mechanisms that can cause this are natural selection, genetic drift, and gene flow. Again, natural selection is the only one that's going to lead to adaptive evolution and responding to changes in your environment. So 23.1, genetic variation makes evolution possible. That we have heritable traits that can vary. When Darwin first described the mechanism of evolution by natural selection, Mendel's work was not known of at that time, and so when he did presented his work on pea plants, that was evidence of these heritable units known as genes. And we now know that genetic variation among individuals is caused in part by these genes or segments of DNA. But that the phenotype is not just going to be based solely on your genotype. It's the product of that inherited genotype and what environmental influences, what other chemicals you might be exposed to. Natural selection can only impact variation that has the genetic component. So here you see two caterpillars that had two different food sources, were exposed to different chemicals, or sorry, it's the same species of caterpillar. They physically do not look the same because the chemicals in their food source were different. So you can have discrete and quantitative characters influence populations variation. The discrete characters are going to be either this or that, while the quantitative characters are going to vary among a continuum. Genetic variation can be examined looking at both genetic variability, gene variability, as well as nucleotide variability. For gene variability, the average heterozygosity is looking at what percentage of loci are heterozygous, what alleles, what genes are individuals most likely to have both a dominant and recessive allele for. For nucleotide variability, we are physically comparing the DNA sequences among individuals. We do see some variation based on geographic variation, which is going to result in differences in gene pools of separate populations. Here you see them in Madeira. There are several isolated populations of mice. The chromosomal variation that's seen among these populations is due to drift. And sometimes we can see this within a cline, which is when there is a change in a trait that is graded that can correspond to some sort of axis in your geography. Mummy chalkfish will vary in an allele for cold adaptation depending on a temperature gradient. And this variation does occur as a result of natural selection. So here you see the mice, you see the different populations of them, and you see some of the chromosomes have fused together. And then here we see a cline. We see the temperature changes in the water from Maine to Georgia, and we see that the allele frequencies are changing along a periodic path as a result of that change in temperature. We can have genetic variation come about due to mutations or duplications of genes. When we have it due to mutation, we've had a nucleotide sequence change. Those mutations cannot be passed along to future offspring unless they happened in their chromosomes that are going to be used to make gametes. If they are just found in their somatic cells, those mutations will not be passed along. We can have point mutations that might have no impact. They could happen in non-coding regions, since there's large sections of DNA that are not used to code for proteins. We could have the mutations have no effect because that gene is coded for multiple times. So it could be a neutral mutation, but we can have ones that can be harmful, that can cause significant changes in a protein that needs to be made. There are times when they can be beneficial. If you have mutations that are of a chromosomal nature that result in multiple loci being either deleted, disrupted, or rearranged, those typically do not work out well. Duplication of DNA is not as significant of an issue because you still have the original and you just added more, so that one is less likely to cause problems. And sometimes when those duplicated genes are added to your chromosomes, they can take on additional functions based on where they are located or by undergoing further mutation. So an odor detecting gene has been duplicated multiple times. We have 1,000 copies of it. Mice have 1,300. Crazy. We don't see these mutation rates as much in animals or plants. There's about one mutation for every 100,000 genes, not too much. We do see them much higher in viruses, and we typically will see lower mutation rates in pure prokaryotes. So they will have fewer mutations take place, they'll have fewer genes before they have a mutation. Sexual reproduction can lead to alleles being recombined. We spent a lot of time talking about this with meiosis. For organisms that reproduce sexually, having that recombination of alleles is going to be critical in producing the genetic differences that are going to allow adaptation to occur as opposed to mutation. So sexual reproduction is a much greater source of genetic recombination. Hardy-Weinberg is an equation that was developed by two individuals to determine at a specific allele if specific alleles within a loci are undergoing change. Is the population evolving at that particular set of alleles at that loci? So a population is something that we need to define. It is a localized group of individuals that are able to breed with one another and produce fertile offspring. The gene pool are the alleles present within that population for all of its loci. If the loci becomes fixed, if every individual within that population has the same alleles and they're homozygous for it, you will not have change be possible because that is the only allele that can be passed along to future offspring. So here we see a porcupine herd and a 40-mile herd, and their ranges overlap. So because of that, they are able to mate and produce offspring, and because they are have their own set of genetic material by producing offspring together, they are able to add variation to their gene pool. We can calculate allele frequencies for a diploid organism by taking the number of alleles that an individual has at a locus and multiplying them by 2 because they have 2 sets of chromosomes. They have a set from mom and a set from dad. The number of dominant alleles at a locus is going to be the 2 alleles for every homozygous dominant individual, and then you have 1 allele for every heterozygous individual, and this would also work for recessives. So if we are dealing with allele frequencies, we represent the dominant allele as P and the recessive allele as Q, and if we take their frequencies and add them together, they should combine to equal all of the alleles frequencies in a population, so P plus Q would equal 1. So here we have wildflowers that are not completely dominant for color. You've got the different genotypes and the amounts of those flowers present. We can determine how many of the alleles are the dominant allele. 320 would be doubled since there are two alleles for the red flowers that are both dominant. And then half of the pink flowers have a dominant allele, so you have 800 dominant alleles. And then for the white flowers, you would double there since there's two recessive alleles, and then add another 160 from the pink since they're heterozygous. And so you get that you have 800 red alleles and 200 white alleles, 800 dominant and 200 recessive. The frequency would be taking the amount of that specific allele and dividing it by your total number of alleles. So 800 divided by 1,000, which gives you 0.8, and 200 divided by 1,000, which gives you 0.2. So allele frequencies within a population aren't going to end up being necessarily 0.5 and 0.5. It's really going to depend on what combination of alleles, what genotype frequencies you have in your population. The Hardy-Weinberg principle says that if the frequencies of alleles and genotypes in a population are static, they're going to remain constant between generations. And this is what we see in a population that is not undergoing evolution. There are certain criteria that have to be in place for the Hardy-Weinberg principle to apply. And these are pretty difficult to achieve. If they are achieved, it can be concluded the population is not evolving at that particular loci. So that when gametes are produced from those individuals, they will get the same alleles, and therefore that allele frequency will stay fixed. Due to genetic recombination and Mendelian inheritance, we should see genetic variation within a population. But if we are not having that variation, we will see the frequencies remain constant within a gene pool. And this could be for a single loci. It doesn't have to be necessarily for all the loci within a population. So if we have frequencies of the dominant and recessive alleles we just calculated being 0.8 and 0.2, the gametes that are produced have an 80% chance of receiving a dominant allele and 20% chance of receiving a recessive allele. So we've got wildflowers, 500 of them, and we have 1,000 alleles, and we're given the allele frequencies for P and Q. We should be able to determine what the predicted genotype frequencies would be. If we know that the allele frequency for the dominant allele is 0.8, we can square that 0.8 value to get the frequency of the homozygous dominant genotype, 0.64. We can take 2 times P and Q to get the frequency, genotypically speaking, for the heterozygous genotype, which would be 0.32. And then we can do the same thing with the homozygous recessive genotype, 0.2 squared. That would be Q squared, and that would give us 0.04. And with that information, if we know those allele frequencies, we should be able to confirm this using a Punnett square. So there you can kind of see the breakdown. Again, it's not because of the frequency of the alleles that we find in the sperm and the eggs, we're not going to see the distribution that we saw back with Mendel. Mendel will say it was a 50-50 chance, but because we know these allele frequencies, we know that we're going to have a much greater number of homozygous dominant plants and heterozygous plants as compared to the homozygous recessive plant. So if there are only these two possible alleles at a particular locus, if you take P plus Q and you square it like you would in the algebra class, you would get P squared plus 2PQ plus Q squared equals 1, where P squared and Q squared are representing your homozygous genotypes, homozygous dominant for P squared, homozygous recessive for Q squared, and 2PQ is representing your heterozygous genotype. Again, this is only true if, or this only theorem is only applicable if your alleles frequencies are remaining constant, if you do not have a population undergoing evolution at that locus. in actuality, these do change. The conditions that have to be met to not be undergoing evolution at a specific locus include no mutations, random matings, no natural selection, and very large population, and no gene flow. And gene flow is due to migration, emigration, and immigration. As I said previously, you can have some loci be in Hardy-Weinberg equilibrium, but others will be evolving. So if we assume that the locus causing PKU is in Hardy-Weinberg equilibrium, what we are saying is that the mutation rate is low, there is random mate selection, natural selection will only act on those rare homozygous individuals who don't follow their dietary needs. There's a very large population, and that migration does not have a significant impact because populations in general have similar allele frequencies. PKU is found in approximately 10,000 births. And so if we calculated that frequency, if we're saying that you have the possibility of getting that homozygous recessive genotype for 1 every 10,000, we can say that Q squared, the frequency of getting that genotype, is 0.0001. Taking the square root of Q squared, that tells us that Q would have a frequency, the allele frequency for this recessive allele is 0.01. And then the P allele frequency would be 1 minus that value, 0.99. With all that being said, now that we know P and Q, we can calculate what the frequency is of carriers of this recessive allele. 2 times P and Q, which gives me 0.0198. And again, this is a frequency, so if I wanted to look at it in terms of percentage, roughly 2% of the population, and this says of the US population, so I'm assuming the PKU occurrence was for American births. 2% of the US population is likely to carry be a carrier for PKU. So you heard me talk about gene flow a little bit on the last section. Natural selection, genetic drift, and gene flow are able to alter your allele frequencies, and this can lead to evolutionary change. Natural selection is the only one, however, that can cause adaptive evolution. So natural selection is going to have an impact on reproduction. Having differential success is going to lead to certain alleles being passed on to future generations in different amounts compared to others. And so an allele that confers resistance to DDT increased in frequency. Once DDT was used in agriculture, those that were susceptible to DDT were not able to survive and reproduce. Those that were able to resist DDT survived and passed on that allele that conferred resistance to their offspring. So we can have allele frequency changes due to genetic drift, and we see this, this is by chance, we see this much more often in smaller samples. We will see that the allele frequencies will fluctuate just randomly. So it would be kind of like, as I use as an example in class, if you had a 50 question test and you miss one, that's not going to impact your grade as much as if you had a 5 question test. So having a random change is going to have a much bigger effect on a smaller population with fewer alleles present overall than a larger population. And so with these smaller populations, we tend to have, or we are more likely to have a reduction in genetic variation because alleles just get lost. So here you've got flowers, you've got your first generation, 5 plants are able to leave off offspring, and that's going to change your allele frequencies. And then only 2 of those 5 plants produce offsprings, and now you have lost that recessive allele altogether. The founder effect is what is seen when some individuals become separate from a larger parent population, and their frequencies are typically different from that of the larger parent population. We can also see this through the bottleneck effect when there is a pretty significant reduction in population size due to some sort of environmental factor. And therefore, the gene pool that is left behind of that population is not um, is not representative or reflective of that original population's gene pool of the allele frequencies that were present. And if that population continues to remain small, we can see even more significant impacts due to genetic drift. So there you see your original population. Some examples of things that could happen would be like droughts or earthquakes, natural disasters, things along those lines. You only keep a few of those individuals from the original population. It is by chance. And if those individuals happen not to have certain alleles, then your allele frequencies are going to change pretty drastically. We saw this with greater prairie chicken. There was a significant loss of habitat in for the greater prairie chickens in Illinois, which caused a pretty significant reduction in their locations of where the populations could survive, and it caused the population amounts to decrease drastically. The birds that were able to survive did not have as much variation, genetically speaking, as the original population, and they had lower reproductive success. And so, by examining DNA from these chickens prior to the prairie habitat loss to after, before the bottleneck and after the bottleneck, we can see the decrease in alleles at particular loci. And when we compared it to other states, prairie chickens, we could see how their allele frequencies remained high, or the different allele possibilities available at a loci, and their reproductive rates were able to stay relatively high as well. So researchers took prairie chickens from other states, used them to introduce these new alleles back to this founder population, and that helped to increase their reproductive success. So there you can see your Illinois before and after, and you can see in the picture with the state of Illinois, just the range, how dramatically it has decreased. And again, comparing Kansas and Nebraska, how their allele frequencies were higher to begin with, but remain there, and their reproductive rates were much greater. So genetic drift is going to have a much more significant impact in a smaller population. There is no rhyme or reason to it. It is not being caused due to adaptation to the environment. It is just random changes. These can reduce your genetic variation, and it can also lead to alleles that are not helpful, harmful alleles, to become fixed, and only those alleles get passed on to potential offspring. So gene flow is when you have alleles move among a population through migration, immigration, or emigration. They could be transferred via fertile individuals or through gametes such as pollen. When we have gene flow take place, it can reduce your variation among populations, and that can lead to a decrease in your fitness. So this particular bird, the great tit, Paris major, allowed mating to occur between different populations on the island. And as that occurred, alleles that decreased fitness for their environment that came from birds coming in from the mainland, immigration, caused a reduction in their fitness, and that allowed them not to be as successful. Natural selection selects for those alleles that are going to increase the likelihood of being able to survive and reproduce in that environment. While the birds in the east that did not have as much immigration did not have an increase in the frequency of alleles that decreased their fitness, and they were able to continue to reproduce at the amounts that they would have expected. So there you can kind of see the population. We see much greater populations in that eastern population where gene flow did not have as great of an impact as it did in the central population. You can have times where gene flow will increase your population's fitness. So it's not one or the other. Um, it can be either. Here you see alleles that help to provide resistance for insecticides have helped to increase fitness. We have, as we as in individuals, have developed insecticides that can be used to target mosquitoes, specifically those that carry West Nile virus and malaria. Populations have had changes in allelic frequencies, have had alleles evolve that are able to confer insecticide resistance to mosquitoes. And when you have gene flow for these insecticide resistance alleles, that's going to help to increase the fitness of that particular population. So gene flow can work in both ways. It is a pretty significant agent of evolutionary change within human populations. But as I've said previously, natural selection is the only mechanism, not genetic drift, not gene flow, that causes adaptive evolution. Evolution by natural selection does involve some chance, but it also involves sorting. The genetic variations are the part that come about by chance. The genetic variations that come about through meiosis, crossing over, synapsis. But the alleles that are more advantageous for that organism to be able to adapt to its environment are going to be the ones that increase their fitness and lead to them to be able to reproduce and pass on those alleles to their offspring. And those are the ones that are favored by natural selection. So, struggle for existence and survival of the fittest are not accurate representations of what's happening with natural selection because there truly isn't a competition among individuals. Reproductive success is definitely more subtle. The fitness part that we've been talking about on these last few slides is going to play a key role. The contribution individuals can make to the next generation's gene pool, comparatively speaking, to those of others. If you have alleles that are going to increase the fitness of that population, selection typically is going to result in those alleles being passed on and those phenotypes being expressed because those individuals are going to be more capable of going through reproduction. When we talk about natural selection, the alleles that are passed on can be in one of three modes: directional, disruptive, or stabilizing. I'm going to show you the graphs, I believe, on the next slide. Directional basically is favoring one end of a phenotypic range. Disruptive is featuring the extremes of the phenotypic range, and stabilizing is favoring the intermediate. So if you have a range of phenotypes, if you have a continuum, you have your original population. Depending on what alleles are going to lead to increased fitness, you can see one of these different selections take place as a result of natural selection, depending on what's going to best benefit that population, what alleles are going to be able to increase the fitness of the population, and therefore those individuals are going to be more likely to be able to reproduce and pass on those alleles. So some adaptations that have occurred as a result of natural selection: cuttlefish are able to change their color in the form of camouflage. Snake jaws are able to be physically extended to help them to swallow prey that's bigger than their heads. Natural selection is going to lead to allele frequency increases that are going to enhance your ability to survive and reproduce. It's going to occur, or you're going to be able to see it more within a population as the organism and their environment match increases. Because your environment is not a static location, it's able to change. Adaptive evolution is not just a one-off process, it is continuous. Genetic drift and gene flow do lead to allele frequency changes, but they do not consistently lead to adaptive evolution because they can both increase or decrease how an organism is fitting with its environment. So sexual selection, we talked about how reproduction is going to be a little bit more, trying to think of a good word for it. It's of course, I come up with it after I mute it. Reproductive success is a little more subtle. Sexual selection is natural selection that's going to lead to success in mating. It can result in having different characteristics between sexes in terms of their physical sexual characteristics, sexual dimorphism. We can have intrasexual and intersexual selection. Intrasexual selection is having competition among individuals, aka typically males for mates of the opposite sex. Intersexual mate choice is when females, typically individuals of one sex, are choosy in selecting their mates. Male showiness can often increase the chances of a male attracting a female. Sometimes that showiness can reduce their ability to survive. Female preferences can evolve over time. It's thought perhaps that if a trait is related to male health, the male trait and the female preference for that trait should increase together. So there you see the peacock and the feathers. We can also have this with vocalizations. Neutral variation, is genetic variation that does not result in either a selective advantage or a selective disadvantage. There are mechanisms that help to preserve the genetic variation that is present in an population. So one of these is diploidy, having genetic variation in the form of hidden recessive alleles. Heterozygotes can carry those recessive alleles that are hidden from the selection process. Balancing selection is when natural selection results in frequencies of more than 2 phenotypic forms within a population. Heterozygote advantage is when the heterozygote has a higher fitness than the homozygotes do. We see this with malaria resistance. Natural selection will typically favor, in the case of this, two or more alleles at a loci. Sickle cell allele will result in mutations in your hemoglobin, but there are also some benefits to it in terms of that malaria resistance. We can also have frequency-dependent selection, where a phenotype's fitness will diminish if it becomes too common. So selection will favor the phenotype that is not as common within a population. And this plays a role in scale-eating fish, those that have right-mouthed and left-mouthed. So there you can kind of see the frequency of the sickle cell allele in populations that are more likely to be impacted by mosquitoes that are able to pass on malaria. And there you see the mouths over time. Natural selection is not able to fashion perfect organisms because selection can only work on the variations that currently exist. It's limited by the constraints that are already present, and adaptations, just like in life, are pretty much compromises. And since nothing is static, you have chance available, you have natural selection, and then you have the environment, and they're all kind of working together and interacting with one another. So you're never going to be able to get everything perfect at one particular time because there's always change occurring.